Thank you. Thanks much to the organizers for making this happen. This has been really fun for me. I didn't prepare a slide to introduce myself, but I like this idea. So my background is also from statistical physics. Mark and I were grad students together. And now I'm very interested in biological problems, and I'm actually not going to be telling you anything specific to biology today, but I hope we can have nice conversations over lunch about the extent to which these ideas apply and are relevant to biology. And I wanted to also give you a little overview of what I'm going to be talking about, I guess, today, Wednesday, and Friday. So the running theme through all of this is going to be information geometry. And today I'm going to start by telling you about why it is that simple models work. I'm going to give partial answers from information geometry. So I'm going to give another introduction to information geometry from a slightly different angle, though there will be a lot of overlap, as I'm sure you'll see. Then I'm going to tell you that model manifolds are hyperribbons for predictive models, but not for curve fitting. So I think this is an important distinction. And I'm going to tell you about how it is that happens. And then I'm going to tell you about how model manifolds are sloppy when viewed at a much coarser scale than that of their components. The next lecture, this is going to be on Wednesday, I think now, is going to be in two parts. So the first, which has a very similar theme, is going to be on why it is that simple models are better, or are simple models better if you don't accept the premise. I'm going to give you a little bit of an introduction into uninformative priors and the idea of Bayesian priors and why they're important. Then I'm going to tell you about Jeffreys geometric prior and an elegant way of understanding what it is from information geometry. And then after building up Jeffreys prior, I'm going to tell you that Jeffreys prior is actually sick for these sloppy models, and that though it has this very nice geometric interpretation, you should think carefully before you use it on a model that has this hyperribbon structure, that has this sloppy structure. And then I'm going to tell you about reference priors and tell you that they may be the answer. That's going to be the first half. And then the second half is going to be a brief detour away from information geometry. And this is to get people on the same page about nonequilibrium statistical mechanics. And I guess the lecture speaks for itself. I'm going to quickly go through the Jarzynski equality and then talk about linear response and derive the fluctuation dissipation theorem. And in my third lecture, I'm going to use sort of a very similar geometric formalism to talk about some bounds for dissipation. So I'm going to give one geometric, a geometric way of thinking about the energetic cost, so how much entropy you produce if you want to leave the adiabatic regime and move a system not arbitrarily slowly, but only slightly slowly. And I'm going to show that this induces a friction tensor metric. This is work by Sivak and Crooks. And then I'm going to talk to you about, since I have a captive audience, tell you about a bound that I'm very interested in for systems that need to be driven autonomously, so that all of the component parts are physically driven as opposed to externally driven. And I'm going to claim that in these systems, there's another entropic bound that uses actually the same Fisher information that we've been talking about and the same information geometry from that. So that's an outline of my lectures. But a goal throughout, so I'd like lots of interruptions and participation. I get very anxious when nobody asks questions. And sort of the two goals that I want to have, in addition to learning or to explaining this specific material, I'd really like people to get some familiarity and intuition for information geometry and how to use it. And specifically, I'd really like you all to leave being able to calculate metrics for lots of different problems. And so in the tutorial that I'll be doing this evening, I'll actually have you calculate metrics for a few problems. Okay. So I'll give a sort of broad philosophical overview of today's lecture too, and then the rest of it will be on the board. So this is an artist's depiction of a cell. I think this is actually for an E. coli, but never mind. And I think what you should notice here is that this is sort of ridiculously complicated. So all these colored blobs here are supposed to be proteins, or RNA molecules, or other macromolecules. They're all sort of precisely molecularly defined machines. They're sort of swimming around and interacting with each other in presumably very complicated ways. And people take these systems and write down models like this. So this is the same model that Mark was showing earlier. And this model makes predictions sort of like this. And there's, I think, two sort of complaints you could have about this approach. One is that this model here is in some ways sort of ridiculously simplified. Surely that's not capturing everything that's going on in here. And it's not capturing everything that's going on for purposes of this cascade, right? So there's almost certainly proteins that are involved in this cascade that nobody knows about. So these ones are known because people found that if you knock one of these proteins out, the cascade stops working. So they're certainly involved, but surely there's other ones too. But I think the other thing you could worry about is that this model with 48 parameters is kind of too complicated for this. This kind of looks like a 2 or 3 parameter curve, right? And so that's kind of the questions that I want to give some sort of an answer to. So you might look at this and say this model is just way too simple. It's not capturing everything that's going on here. Or you could say this model is way too complicated. And I think both of these concerns are valid, but just as a matter of fact, if you write down these models and fit them, they do a pretty good job of capturing the data. And they don't do a lot of overfitting. So if you looked at this data, you'd probably say that those wiggles are noise, and your model agrees. It's not sufficiently flexible to hit those data points that are all over the place. This is for this complicated biochemical model that we don't really have a great idea for how to understand, I would say. It's an open question. But we could have asked a very similar question about this system. So these red and white things here are water molecules. And this big funky shaped object is a molecule that's diffusing around in it. And all I want you to take from this is that this is also ridiculously complicated, right? So if I really want to understand how this molecule is moving around, I would need a very detailed model, right? I'd need to know how it forms hydrogen bonds with all these water molecules, how water itself is structured, and how it breaks through this structure, and a lot of details about water and about this molecule. It would probably really need to be quantum. So water is quantum mechanical, and you get wrong answers if you ignore this. But if you're only interested in things that happen at sort of relatively long timescales, you can use the diffusion equation. So here I'm representing the density of the particles, and here's a diffusion constant. And somehow all of these details about sort of how water interacts with itself and how it interacts with the molecules, all of this just gets compressed into this one parameter d, right? So we have this very complicated system, and all these other details, the ones that don't affect d, are in a precise renormalization group sense irrelevant, but also in our everyday use of the word, right? That there's lots of details that don't influence d, but they're irrelevant. And one last question, which certainly seems related, is why is physics universal? So in work that I'm not going to be telling you about much, I have collaborators who are very interested, who do beautiful experiments really with membranes. So what you're looking at here on the left is a synthetic membrane. So it's composed of a few different lipids. And one of them is fluorescent. And if you lower it down in temperature, it'll actually phase separate. So if you were to lower this in temperature just a little bit more, it would look like you were looking at a phase of the moon. So one half would be bright and one half would be dark. And as you get close to the critical point, you see these large fractal structures that are much larger than individual molecules. So individual molecules here are a nanometer, and this structure is about 10 microns across. And that's a synthetic membrane, but this is a cell-derived one. It has another fluorescent probe in. And I would claim to you that if you looked at the statistics of these fluctuations at these large length scales, you wouldn't be able to tell which was which. So somehow there's emergent, interesting structure here, but it doesn't depend on too many of the details of the model. And in fact, if I do this very simple Ising simulation, which I'll talk about in more detail, that will also have quantitatively similar structures. Okay. So an outline for today, a more specific outline. First, I'm going to tell you about how the widths of the model manifolds are sloppy, so they have this hierarchy of scales from interpolation. Then I'm going to talk about how the metric, the Fisher information, which I'm going to switch to calling it G, as I coarse in my observables in time, it's going to become sloppy with the most stiff directions, so the most relevant directions, describing diffusion and drift. And then I'm going to talk about sloppiness in the metric on coarsening in space. And I'm going to show that when you do this, you recover this 2-parameter Ising model from a many-parameter Ising model. Okay. Okay, I did it the bad way. Okay, so I'm going to start talking about interpolation, and to do this, I'm going to start with the same model that Mark introduced and that we're going to see quite a bit more of. So y of t, I'm going to normalize it slightly differently. Like this, so it's just going to be a sum of exponentials, the sum of n exponentials like this. And as before, so sorry, I'm going to also change from d to x. And the probability of observing a particular x given model theta is going to be 1 over 2 pi sigma. It's going to be like this. So my model is going to make a deterministic prediction, and then I'm going to assume that the data I actually see is Gaussian distributed around that prediction. And let me draw this out. So here's t here. Here's y here. All of my models are going to start at 1. And if I have some actual data taken at several time points, so here's t1, t2, and t3, this is going to look something like this. And the only constraints that I want to place on my parameters here are that theta mu are greater than or equal to 0. So I don't want to allow negative ones. And then I can have some theta best fit, which looks something like this. And now what I want to start by doing is actually deriving the metric for this model. So we've done it before, but I'm going to start from a slightly different tact, from a different angle, and that's from the KL divergence. And I want to consider the KL divergence between two nearby models, P of theta and P theta plus delta theta. And remember that we can write this as the expectation value of log P theta of x minus all on the probability distribution defined by theta. When I expand this out, I get so where this now is shorthand for log of P theta of x, right? And d mu is defined to be d by d theta mu. Delta theta mu. There should be a minus sign here, right? So 1/2 here. And now we have like this. So what do we do with this term here? Yeah, this term is 0, right? So Mark showed this in the last lecture, right? This is 0, and can anyone say, and so we talked a little bit about properties of the KL divergence, but can anyone say in words why this kind of has to be 0? Why it would be very troubling if it weren't? Yeah, so we talked about how the KL divergence between any two models, close by or not, has to be positive. And if this weren't 0, then I could choose some delta theta that was sufficiently small that we could ignore all these other terms, but was such that this was negative, and then I could have two models which have negative distance. And that seems very problematic, right? That means that I could write down this model, P of theta, and I'd be able to fit it better with a different model. Right, so this doesn't make any sense, so this better be 0. Now, the other thing which we talked about is that this term has to be, well, what do we know about this term using similar arguments? Yeah, it better be positive definite. And in fact, I can write this as 1/2 theta mu. I'm going to write this like this, and one of the things that I'd like, or that we should do as an exercise tonight or this afternoon, is showing that G mu nu, the Fisher information, we can either write as minus mu d nu L of x, or as mu L of x d nu L of x. So this is an exercise that we'll do tonight, but once it's written in this form, you can see that it has to be positive semi-definite, right? Because this is a covariance in any direction that I move in this. I will be positive. So this is a covariance matrix. That means both that it's symmetric, or both that it's positive definite, and also that it transforms like a metric. So let me write that out. So if I make a coordinate transformation from theta to theta prime, my new matrix G prime, which I'm going to write in alpha beta coordinates, is going to be written as sum over mu and nu theta prime alpha theta prime beta. And this really follows from working through this. This is another good exercise. But what this means is that this really transforms as a metric should. And so we will be treating it as a metric for the rest of this. I also, just to think a little bit more about this KL divergence, it's often called a divergence rather than a distance. And I actually think it's closer to a distance squared than a distance, right? So if I go to linear order, there's no divergence between two close by models. But as I go farther than that, I get a distance that looks like this. If I were to include the third order term, I'd have contributions that look like derivatives of the metric plus Christoffel symbols. And generally, it looks at least locally like a distance squared. That's correct. So so for models for which there's odd terms in this, and this is the only odd term that has to vanish, and the other odd terms will be strange because they're asymmetric as I move from one direction to the other. Yeah. Well, so whether there are some cases where this is exactly what you want, but it is not. It's not a distance. And you can show various other problematic things. So it doesn't satisfy the triangle inequality, for example. Okay. So I'm just going to briefly, yeah. Yes. So here, what I want is this as an expectation value over theta. Oh, I'm sorry. Yes. Yes. I guess I wrote it out in full here, but I always want to have Einstein summation. Yes. Okay. So I want to actually go through and calculate the metric again for nonlinear least squares. So nonlinear least squares is problems of this form where I have a deterministic prediction, and I'm going to blur it with Gaussian error of this sort. So log log p of x given theta is going to be some constant plus 1 over 2 sigma squared sum over I xi minus yi of theta squared. This is getting small. And d mu log so I'm just working through, and I'm going to actually just calculate the metric according to this, and then we're going to use that to show that when I look at the metric of this, it's going to be sloppy, so that people don't get lost in the math here. Now I want to sum over I dyi d theta mu. Get rid of the 2 here. Xi minus yi of theta. Right? G mu nu is going to be equal to 1 over sigma to the 4th, sum over I and j. And now we have dyi d theta mu, dyj d theta nu. So remember, this is the Jacobian. And here we have like this. And how do I simplify this? So this part here is all deterministic, right? I can take this out of the expectation values, or this does not depend on x, but this part does. So these are Gaussian distributed around y, right? So this is going to be delta ij, right? So I'm assuming that my different time points are uncorrelated with each other. And then sigma squared, right? So that's just the size of the fluctuations in each one. And that means that I can write g mu nu as 1 over sigma squared j transpose j, where this is the same formula that Mark derived earlier. So just giving a slightly different derivation. And there's one more thing that I'd like to notice. Remember that, where is it here? When I do a coordinate transform from one coordinate system to the other, I end up multiplying by this Jacobian factor here. And what that means is that if I instead of using these coordinates, if I use as my coordinate systems the y's themselves, the yi's, then my metric is Euclidean. So let me draw that. So here's y3. y2 and y1. So this is the plot that we were seeing before. And I'm just going to draw it as it was before. So the one we were looking at before was in 2 parameters. But now I'm actually going to draw this as if n were large. So I'm overfitting this, right? If n is large, I certainly can't extract all of these theta mu's from data. But I want to draw it nonetheless. This is what it's going to look like. And what I want you to notice is you could see these first 2 directions even with just 2. And there's one very long direction. There's one thinner direction. And then there's one very thin direction, right? And if I want to put my error bars on this, there's something like this. And this is the yardstick by which I measure Fisher information distances, right? So if I put 2 parameters on here, and I want to ask how far apart they are, at least if they're close, where I can write ds squared as right. So for close by models, I can do this. What I want to do is put them in here and just see how far apart they are. And distance in this metric, as Mark was saying, is really measured in standard deviations, right? So this is measuring how far apart these models are. And what this means is that any direction which is thinner than a standard deviation is sort of quantifiably not distinguishable from typical data, right? So this long direction, at least with the error bars that I've drawn, you could pick out where you are along it and distinguish whether you're here from here. This one, maybe you get a little bit of information, but not a lot. And in this third direction, you're really getting no information at all. So before stopping and going into detail why it has this structure, I think it's good to just absorb what this structure means, right? That as I move from here to here, I'm changing my parameters very dramatically, but in a way that doesn't make a big difference to the predictions. So Mark is going to be talking quite a bit about what these edges correspond to. But for now, suffice it to say that as I move from one edge to the other, I'm moving some of my parameters from 0 to infinity. And even as I do that, I get almost no change in the behavior. OK. So to think about why it has this structure, I want to think about what this means. And one thing which should maybe bother you is that even though I have an infinite number of parameters, or a large number of parameters, and I'm only trying to fit 3 data points, as I've drawn it here, the best fit does not fit all 3 of these, right? And I don't know, so why is that? Why can't I fit this? Yeah, so exponentials never go up, right? So because of this Gaussian error, my data point can go up. But even though I have this very high parameter model, it's not capable of fitting it. And in fact, the exponential is quite structured, right? So the first derivative is always negative. No matter what I choose for these theta mu's, you can tell me a lot more about it than that, actually, right? That the second derivative is always positive, the third derivative is always negative, and so on and so forth. And this function, in fact, is analytic and smooth and quite constrained by all these things. So what does this mean for fitting? Here's T1, here's T2, and here's T3. Here's 1 here. I first just want to think about the behavior before I've actually taken data and think about the accessible model space. So before I've taken any data, get the color right, I can really have anything in here, right? There's no data point here that I couldn't fit perfectly. But now let's imagine that I take a slice through this manifold at a particular value of y1. Sorry, of y3. So I only want to consider models that fit through here. And on this plot, that corresponds to something like constraining myself here, and only looking at, well, it's hard to draw, but some little slice through this, right? And I say it's a little slice because if I look at the behavior that's allowed now anywhere else, it has this form. So I can't ever get below this on this side, right? And I can't ever get above this on this side. It turns out you can convince yourself that you also can't be above this line, where this line is what's given by just a single exponential. And you can try and convince yourself of that. And so just taking that one point, which corresponds to taking a cut through this, really constrains the available behavior of the model. And we can do the next cut, which I'm going to put here. And let's go for somewhere in the middle of this behavior. So somewhere let's say here. And this also constrains the model quite a bit. So you can convince yourself that this actually, it's no longer okay to start here, because if I started here, I'd need to continue on like this forever. Something like this. I've not drawn this very well, but the point is this further constrains things quite a bit. And what that means is that when I take this slice through the model, corresponding to fixing parameter 1, let's see, so that's going to be something like this. And now I'm just left with this very small range here, and that's this width here. Let me just erase this. So if I plot the model widths, so here's WI. When I plot them for this, experimentally what I find is that they're evenly spaced in log. And the spacing here corresponds to the radius of convergence of these lines that I'm drawing, divided by the time difference between these models. So the closer spaced I have my time points, the farther spaced these different, well, these eigenvalues of the Fisher information will be. Any other questions about this? Okay, so next I want to move on to a model of diffusion. So here sloppiness really came about because some of the data was able to constrain the other data, right? So once I gave you this data point and this data point, I didn't need to tell you the model to tell you what this data point is, right? And Mark was arguing before that this is sort of important for having a predictive model, right? If your data isn't sufficient to constrain other data points, it's not really a predictive model. And if you're in that regime, probably it's already predicting some of the data points you've already taken. So now I want to turn to another way that sloppiness can arise, which is when you take data at a time scale that's much longer than that at which the model is defined. So now I'm going to erase this. And as I'm erasing the rest of the board, I'll put the model up. And what I want this to be is a stand-in for water, right? So water is extremely complicated, and I'm not going to try and write down a reasonable microscopic model for water. But instead, I'm going to write down a one-dimensional hopping model, and we're going to show that this one-dimensional hopping model, which is not as complicated as water, but is still too complicated for diffusion, that when you coarsen it, it's going to become sloppy. So here's the model. It takes place on a one-dimensional lattice. And time is also going to be discrete. So in every time step, every particle is going to hop according to a kernel that we're going to parameterize. So here's theta 1. Here's theta 3. Theta 0. So as I've drawn it here, there's 7 parameters to this model. Probability is not necessarily going to be conserved, or particle density is not. So if these don't sum to 1, then every time step particles are going to be created or demolished. And we can write this down in the following way. So this is I in this direction. yi at time t is going to be equal to the sum over mu of yi minus mu of t minus 1. So our data here is going to be the density of particles. And imagine that I have a lot of particles, but not a very good way of measuring them, so that my errors are going to be Gaussian distributed, just as they were before. And I want to start with initial conditions that yi of 0 is just going to be delta I0. So I'm going to start with all of my particles sitting at the origin. Does this make sense? Yeah. Sorry? Oh, I'm sorry. Indeed. Okay, hopefully it didn't make sense before. Okay. So, do you guys buy that this is a reasonable stand-in for water at the microscopic level? Okay. Well, we'll see if you believe that the results should hold for a microscopic model of water. So let's first calculate the metric after a single time step, because this we can actually do. After one time step, well, delta I of 0 times theta mu is just going to be theta I, right? And we can easily calculate the Jacobian for this. So what is the Jacobian here? So this is, let me call this the Jacobian after one time step. So this is dy I of 1 d theta mu. Yeah, so this is just the identity. And we can also easily calculate the metric here. So this 1 over sigma squared is going to carry through in all of these. Sum over I, delta I mu, delta I nu. And this is just 1 over sigma squared, delta mu nu. Erase this a bit. Okay. I'm still assuming a Gaussian error bar. So you could imagine doing something more systematic and saying that these are probabilistic and yeah, and you'd get qualitatively similar results. So now I'm going to plot the eigenvalues of the metric. Log lambda. And this is unambiguously not sloppy, right? This is the identity. All the eigenvalues are the same. This is t equals 1. And I don't know, this kind of actually makes sense. So I don't know, why should this not be sloppy? So I claim this is a stand-in to looking really at microsecond, or sorry, I guess picosecond snapshots of something moving around in water, right? And if you could do that, you presumably really could understand quite a bit about the interactions of this molecule with water, right? If you're really allowed to look at the microscopic motion at this scale, you know, in this model, when I measure the density here, I'm really very specifically measuring theta 1. And when I measure the density here, I'm measuring theta 2. And there's no mixing between them. There's no hierarchy of parameters. Each parameter is measured independently. And this is kind of like, now that I'm thinking about it like this, like experimental design, that in this experiment, I'm measuring each of these parameters separately. OK. But now imagine that you can't do this, and you have maybe you're looking at these are fluorescent molecules, and they're moving around in water, and your microscope can't do picoseconds. It's much slower than that, and so you can only see after several time steps. And yi of t large, you actually know from the central limit theorem, or I will tell you from the central limit theorem, that as time gets large, the distribution of these has to be given by a Gaussian. So let me draw that. Here's y. Here's the origin here. Here's y of t here. I'm taking a not very specific continuum limit here. But I want to emphasize a few things here. So this Gaussian, I claim, has 3 parameters. So this is y naught of t. There's a width. Sigma of t. And if I haven't chosen my thetas to sum to 1, then if they don't sum to 1, then the number of particles will exponentially grow, right? Okay. So let me show you how this comes about. So this calculation was easy for the metric after 1 time point. And numerically, it's still easy after many time points. But it's going to be a zoo to write out like this. But I will, yeah. Yes. Yeah, so if these parameters don't sum to 1, that's quantified in R. Actually, sorry, let me write out one more thing. So in the continuum limit, this is going to be described by dt rho equals d grad squared rho. So this is the density, the continuum version of y. And this is what I had down at the beginning. There's going to be v grad rho. So if my hopping model is asymmetric, if it's more likely to hop to the right or the left, that's going to be quantified in this v. Yeah. Wait. I'm sorry. Yes, this is rho. I'm sorry. This is I. And this is indeed y of t. That's R rho. Okay, and as time proceeds, these eigenvalues will separate. So if I waited a long time, which of these parameters would be the easiest to measure? This is maybe a little bit of a trick question, but I mean, I claim it's this R, right? That if you waited for a long time, if you had some particles whose density wasn't conserved, the longer you waited, the easier it would be to see that, right? And indeed, make sure I get my, no, that's green. The longer you wait, the higher this gets. And this the eigenvector that corresponds to this eigenvalue is just the sum of all the parameters. So this is really measuring R. What's going to be the next easiest to measure? So this is a good guess. It's actually the drift. So if this is drifting, then the longer you wait, the easier that'll be to see. And that would be in blue. Let's call that V. The next one, I guess I've given away, or you've guessed. This is the drift. And actually, drift is marginal in the RG sense. And because of the way I've defined this, in this problem, it actually slowly decreases. But this stays roughly the same. So the longer I wait before my measurement, it doesn't really matter for measuring the diffusion constant. And that's the last parameter with a name. Put the name up here. And all of these will spread out. It turns out that they'll decrease with their RG eigenvalue if that connects. We'll talk a little bit more about it in the context of the Ising model. But first, this really quantifies why these parameters don't need names, right? So in the case of water, presumably, if I could really look, there is some parameter that measures whatever this parameter is measuring. So actually, any thoughts on what this measures? Yeah, so it turns out that you can think about these as measuring cumulants of the eventual distribution. So this R is measuring like the 0th cumulant. V is measuring the first cumulant, so the mean drift. D is measuring the second cumulant, the diffusion constant. And then this is measuring the third cumulant, or the skew of this distribution. And because of the law of large numbers, this skew is getting proportionally smaller every time step. And this is measuring the fourth cumulant and the fifth cumulant and so on and so forth. And they each get progressively harder to measure. Yeah?
You know the spiral form, then you only like 4 theta.
Yeah, so if I, right, so if I knew this from the beginning, I could try and find a simpler description than this. And, well, in some sense, that's what this is, right? That's because you know that there's only 3 terms which could possibly enter into this continuum description, or because only 3 do, you'd never use this 7-parameter model for anything. You'd just start with this 3-parameter model. What was I going to say? Yeah, so I'd say another way to think about it is often due to symmetries, right, R is 0, right? So for many problems, particles don't create themselves, and particle conservation is a reasonable thing to assume. And that lets us drop this term. Remember at the beginning I showed you the diffusion equation without this second term. And that's also common, right? That if I have a particle that's just diffusing in space, because that's symmetric under inversion of the spatial coordinate, maybe I can set V to 0. And then there's just the diffusion constant to worry about, but I can't set the diffusion constant to 0, right? So if the second cumulant is 0, then all higher cumulants are going to be 0. And so I think this gives another way of thinking about why you've never seen a model with this parameter in it, and it doesn't have a name. Okay, I think this is all I want to say about the diffusion equation. Any more questions? Yeah.
Can a linear microscopic model give rise to a nonlinear map?
Sorry, say again? So.
A linear microscopic model give rise to a nonlinear microscopic model?
I'm not sure. Yeah, this sounds reasonable. Yeah. So the question, since I have the microphone, is that can you have a linear microscopic model that leads to a nonlinear macroscopic model? And I guess the answer is yes. That if we believe in quantum mechanics, then it's all linear at the bottom, and somehow lots of effective theories are not. And actually, another thing to say is that these constants here, so this one and this one, curiously, are linear, but this one is not. So maybe that's a more direct answer. I don't have a good explanation for why V and R are linear. Yeah.
So there's another way. Diffusion equations are listed as the row.
Yes. That's true. Yes. So maybe this doesn't count for what you're saying. Yeah, so I was saying that the diffusion constant is nonlinear in the microscopic parameters, but this is still a linear parameter, I think, by what you're saying. Okay. So I want to move on to talking about the Ising model in the last half hour. How many people are familiar with the Ising model? Okay, so I will I will start from the beginning, but hopefully this won't be too slow. So I'm going to be talking about the two-dimensional Ising model, and actually one of the things that we'll do in the tutorial this evening is talk about the 1D Ising model, and you'll actually calculate the metric for the 1D Ising model. For the 2D Ising model, at least with to my knowledge, there's no exact way to calculate the metric, but we can still estimate it with Monte Carlo. So I'm going to be telling you those results and showing you how we calculate it. And then giving scaling arguments for what we find. So the Ising model takes place on this two-dimensional lattice, and there's going to be spin variables that sit at all these sites. So +1. I won't fill this all in. And now the data that we're going to be trying to understand our parameters with is actually going to be the whole ensemble of spin configurations. So if you want to try and understand what the parameters are, I'm going to allow you to look at the entire configuration and do whatever sort of analysis that you want on it. And we're going to try and find the Fisher information for that. So the probability distribution for a particular configuration is going to be given by 1 over z x minus h of x. So I'm going to assume that beta is equal to 1 for this. And furthermore, I'm going to write h of x in the following way. It's going to be a sum over parameters with a minus sign, beta mu phi mu of x. So these are going to be my parameters. These are called conjugate variables. And if I tell you what the conjugate variables are, that's enough to define the model class, or the model that I'm thinking of. So I'm going to have phi 0, so this is going to be a mu index here. So this is going to be just the sum of all the spin variables. And then I'm going to have phi alpha beta, where this whole thing is a mu here. This is just convenient to write like this. That's j plus beta. So this looks a little bit confusing, but if you're familiar with the usual Ising model, I can write it in the following way. So phi 0, sorry, not phi 0. Beta 0 is just going to be beta h, right? Yeah, h or beta h. And what do I want to do for the theta alpha betas? Yeah, so theta 0, 1 equals theta 1, 0 equals beta j. And all the other ones are going to be 0, right? So this is the usual Ising. Just to write that out, so these are my thetas. It's a little bit confusing which ones you want to include to ensure that you've covered all of the local interactions. But what I want to do here is calculate this metric in all of these other directions and try and justify the use of this two-dimensional Ising model. Two-dimensional, but also two-parameter Ising model. Okay. So the first thing we need to do is actually calculate the metric. And we're actually going to calculate the metric, the Fisher information, if we could see this entire probability distribution, right? So I'm not going to specify what the algorithm is to infer the parameters, but we don't need that to calculate the Fisher information and to know it's a bound. So g mu nu is going to be minus g mu d nu log z. I'm sorry, I've gotten ahead. D mu d nu log p of x, right? And I can write this as, well, log p of x is going to be h of x, or minus h of x, plus d mu d nu log z, and this is just a constant, right? So this is going to be we'll change that. We okay with this? Okay, so how do I simplify this term? So this term is linear in the parameters, right? So this is potentially nonlinear in the spin variables, right? Through this sort of term. But in the parameters, it's linear, right? So when I take two derivatives, this goes to 0, right? So this equals d mu d nu log z. And this is another good exercise. This I can also write in two ways. So one as d mu phi nu, and one as phi mu minus phi mu phi nu. Have people seen this before? So this is sort of the magic of partition functions. I won't derive it here, but it's just a little bit of algebra from the definition of this, which I guess I also haven't written down. As the sum over of this, and this is a good exercise. And this is the metric. So it's a connected correlation function of the conjugate variables. And the thing that makes this very convenient is that we can actually just calculate this from Monte Carlo, right? So if I run a Markov chain Monte Carlo, I can write down phi mu and phi nu for every term, or for every microstate that I get, and I can just compute all of these things and calculate the Fisher information. And over here, I will actually just tell you what that gives. Yeah, so in this coordinate system, actually, so this writing the Hamiltonian as linear in the parameters is kind of like a coordinate system. And in that coordinate system, the next term is actually the Christoffel symbol with all lowercase indices, which I don't think should be obvious, but that's coordinate system dependent. So that's not true of nonlinear least squares, for example. Okay, so here's log lambda here. And just numerically doing this, we see this. So there's two directions which really seem to distinguish themselves, and a whole bunch of other directions which do not, but which don't seem to be spread out in this sloppy way. And actually, looking at this definition, the very first term, so another thing you can maybe see is that if we calculate this along the line of the Ising model and zero magnetic field, which is what we did, any term in the Fisher information that mixes the zero direction with the alpha beta directions is going to be 0. So this eigenvalue is actually GHH, or G00. That is an eigendirection. And looking at this, this is the magnetization, the magnetic susceptibility. I'm going to put a proportional sign there because the magnetic susceptibility is defined with, well, anyway, this is the susceptibility. And if we only had these two parameters, so if we only had theta 0, 1 and theta 1, 0, and we didn't have all these other parameters, then this would be the specific heat. Because we have all these other parameters, they get mixed in. So it's not the specific heat, but it still diverges like the specific heat. So this I can't write like this. It's mixed into all of the different eigendirections. But it diverges like the specific heat. And this goes like L squared times the correlation length to the 7/4. This is an Ising exponent. And this goes like L squared log of C. And then what I'll just put as a mystery for now, all these other eigenvalues which don't have names, they scale like L squared. So we called this a hyperrod. So there's two directions which are long and extended. and distinguish themselves, but the other ones don't contract onto this hyperribbon structure, right? They have a scale, and that scale is just the system size. So we were quite perturbed by this, yeah. So the specific heat would be d energy d beta, right? I can't write it in terms of these, but I'm sorry, that's true. It would be G0101 plus G0110 plus, right? So it would be the next eigenvalue, yeah.
All the four
yes, yeah. So we were quite perturbed when we saw this, but in retrospect, we shouldn't have been. So I don't know, can you interpret, like maybe why do we see these not as being spread out in log, but all being sort of concentrated here? So I would say these ones are universality, right? That these ones are saying, you know, it doesn't, yeah, well, I'll talk a little bit more about these, but somehow these you're not measuring from microscopic configurations, right? You're seeing how far you are from the critical point, and you could look at that from a distance, and you don't need to look at the detailed configurations, and you could tell me how close I am to the critical point and how magnetized I am. But these ones, I would say, if I give you a microscopic configuration, you really could tell me whether it was a nearest neighbor or a next nearest neighbor that's defining how far away from the critical point you are, right? That you know, they would have a very similar coarse-grained effect on phase separation, but microscopically, you could say, well, you know, do these tend to be more correlated or do these tend to be more correlated? And from that, you could try and infer all these parameters. Sorry? Well, okay, so I don't know that I'd be able to specify how exactly you should infer what the parameters are, but you probably could do it, right? If you looked at enough of these microscopic configurations. Okay, so so what we wanted to do next is think about how we could coarse grain this by analogy to the diffusion equation, where we coarse grain by blurring in time. And what we're going to do now is something akin to decimation for RG, if this connects. So let me draw this out here. Sorry? Okay. So one other thing, if I look at this like this, d mu d nu log z, this is d mu d nu of the free energy, and the free energy is extensive. So this should be extensive, and so maybe it scales like the system size, and there's no other divergences, and exactly. So it turns out, and what I'm going to show you in a second, we can trace these to sort of irrelevant parameter combinations. So d mu d nu log z, I can write as minus d mu d nu f. And f, I can write as f singular plus f analytic. And these two large directions, we trace to second derivatives of the singular part of the free energy, and everything else we trace to derivatives of the analytic part. And I don't know what the analytic part looks like, but if it's uninteresting, then we'd get something like this. Okay, so to coarse grain this system, what we're going to do is just decimation. This is not really square. So what I've been telling you is imagining that I can try and infer the parameters by looking at all of the spins together. Yep. So one thing so
for example, then you have compartment period.
maybe I'm say again. So
Suppose your second nearest neighbor interaction with the Ising model.
yeah.
You can build your Hamiltonian with different theta than just so things and the nearest neighbor.
Yeah.
Theta 0, 2 and theta 2, 0.
Yes.
Would you still have only two relevant directions?
Yeah. So actually, I should be more clear as to what we actually did, which is we calculated the metric along the line of the usual Ising model in zero magnetic field at the critical point, but we looked in all these other parameter directions. So these eigenvalues are for the Fisher information that's not just of nearest neighbors, but of next nearest and third nearest and fourth nearest, and including 12 terms. So is that answering your question? So we didn't actually do simulations and calculate the metric at other points, but we calculated the metric in these other directions. And yeah. Okay, so X, what I've been showing before is looking at all spins as observables. But now I want to define coarsened spins, so okay, better get this right. So this is x1, and I want to define x1 to be the observable at level 1. And now what I'm going to want to try and do is infer my microscopic parameters, but only looking at these spins as observables. Right? So this is like decimation, and in the usual RG scheme, what I then want to do is write down a coarse-grained model. So I'd write down a new Hamiltonian that just describes the configurations of these pink spins. But now instead what I'm going to do is actually calculate the Fisher information of the microscopic model of the bare parameters, but using just these as observables. And level 2 is going to have this. Let's see. Sorry. Put a tilde here too. So tilde designates coarse graining. And I can keep going like this. So we're now going to do algebra to show how you actually calculate this with Monte Carlo. And then I'm going to show you the results of the Monte Carlo. So give x tilde n. I'm going to write in this particular way. So I'm going to write this as the coarse-grained partition function of x tilde n divided by the bare partition function. And this is just going to be the partition function that's constrained to coarse grain to this particular x tilde n, right? So if I'm only allowed to look at these pink spins, then there's a lot of microscopic configurations that will contribute to each macroscopic configuration, right? So z tilde of x tilde of n is just going to be the sum of x at coarse grain to x tilde of n. Of x of minus h of x. So do we agree that this is, one way of writing the probability for x tilde of n? Okay, so now I can write the Fisher information with these as the observables. So g tilde mu nu is going to be mu d nu minus d mu d nu log z, right? No, sorry, this is right. Yeah? And this we're going to calculate in a very similar way. So I just want to define one more expectation value of a microscopic observable, so an observable that lives on the microscopic lattice, but constrained to configurations which correspond to this particular x tilde of n. So I'm going to write this as a curly bracket of some observable which lives really on the microscopic configurations. But constrained like this. So this is just going to be 1 over sum over x in x tilde of n. So O of x times yeah. 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 So I'm sorry, I didn't define these very well. Let me write out what I plan to write here. So this is x1 is Sij for ij that are pink. Right? So my data is just going to be the spin configuration, the spin values, but only the ones that are pink in this plot, right? So this is what you'd do if you wanted to do RG with decimation, except there you'd actually write a Hamiltonian that lives on these spins. And here I'm just going to calculate the Fisher information that lives on the bare parameters. X2 is yeah, so it's just going to be the same thing, but for ij green. Okay, this is confusing the way I've drawn it, perhaps, that because of the invariance of the, or because of things about the square lattice, I can have them be non-overlapping, but I could also have these be right, so it could be a subset of the pink ones. Yeah. So, okay, just to like. Exactly. So in this scheme where I'm drawing x's, x3 is going to be like this, and yeah. Yeah, so this is, yeah. Okay, so the other thing is that I can actually calculate this with Monte Carlo, right? Well, roughly what I need to do is I need to start with this. I need to pin these spins x tilde that I want. And then I need to do Monte Carlo on the rest, and I just need to reject any sweep that tries to, or any move that tries to change one of these. And if I just do detailed balance on the remaining spins, I can actually calculate this ensemble, and I can calculate these observables like this, right? Is that clear? Yeah. So this is an x, Because my Hamiltonian's only defined on the microscopic ones here, right? Ooh. Yes. So I want. Yeah. So I want to sum over all the configurations that coarse grain to this x tilde.
Okay.
Yeah. Yeah. All of the x's which are not pink, I integrate over. Okay, so maybe you'll believe that in this notation, this is another good exercise, that d nu log z tilde of x tilde n, I can write as phi mu phi nu minus phi mu phi nu where this term here means that I need to do it from the same x tilde n, right? So with this, the other thing that I need is that if I take the expectation value if I write it like this, then these inner brackets are redundant. So this equals phi mu phi nu. Can anyone argue this? Yeah, so I mean, the way I calculate this is I first calculate a coarse grained configuration, and then I average phi mu phi nu in that. But that's just the same as doing this. But if I put brackets around this guy, that's very different. That's something that I can't write without the curly brackets, right? But it's still something that I can calculate by doing this Monte Carlo scheme that I described. And in fact, g tilde n mu nu I can write as mu phi nu. Can you guys see this over this here? So this looks just like the uncoarse grained one, except that now when I calculate the connected part, I need to do it from two different configurations that are randomly chosen from the ensemble that coarsens to the same x tilde. So do we see roughly how we could calculate all these terms? So this term we've already talked about how to calculate. And this one we can calculate by generating these ensembles that coarsen to the same x tilde. So with that, I'll actually just show you the results from this Monte Carlo.
So again, the bracket is the coarse grain.
Yeah, so
The curly brackets and the fine brackets.
exactly. So this this has an x tilde n and an x tilde n. So these x tildes are the same, and this is an average over x tilde n. Yes, all the curly brackets have x tilde n's, yeah. Okay. Oh, and I need this to go down. It's going to be a close call. Time. cool. Okay. So this is what the spectrum looks like. So this is what I drew on the board. This is the magnetization, and this is the specific heat that's measuring changes in T minus Tc. And as I coarse grain, these two eigenvalues stay relatively fixed. You can convince yourself that they can't possibly grow, right? Because I'm only throwing out data, and if I throw out data, you know, I can if there was a scheme to infer this better after I've thrown out data, then I could use that scheme at first and just throw out some of my data. But these two directions which are relevant don't contract, right? So one way to say that is if I tell you to look at these and infer the magnetization, infer the magnetic field and the distance to the critical point, if I gave you blurred configurations, this would cause no problems, right? So if you were an Ising expert, you could look at these and tell me how far I am from the critical point, and it wouldn't help to look at the microscopic details. But these other directions, which are measured from the analytic part of the free energy, or which you're really measuring by looking at microscopic configurations, they contract as you coarsen. And in fact, these contract with their RG exponents, though the relevant ones don't grow. And I guess I'm over time, so yeah.
Shouldn't the number of eigenvalues increase as you coarse grain?
Yeah, so this is, so I'm always looking at the same 12 parameter microscopic model.
You just reject the other.
Well, so the Fisher information is always going to be 12 by 12, just because that's the number of parameters that I keep track of. But in some sense, we don't ever actually write down the coarse-grained Hamiltonian. So this, modulo Monte Carlo stochasticity and such, this is the exact Fisher information after coarse graining, but it doesn't tell you how to build the Hamiltonian of the coarse-grained model, if that makes sense.